Good afternoon, everyone. Thank you very much for joining our webinar series from the European Sequencing Store from Roche. Today, we have a very important speaker, and I'll introduce myself. My name is Marcos Michalatos. I'm the International Business Leader for the KAPA sample preparation reagents. Our portfolio involves tools, high quality, high performing target enrichment, either with a KAPA HyperCap workflow, that is hybridization probes-based technology that comes with custom panels, KAPA HyperChoice, KAPA HyperExplore probes, the latest KAPA HyperExome V2 Probes for whole-exome sequencing, as well as other catalog panels for hereditary genetics and oncology. It is based on the high-performing and high-conversion library operation kits from KAPA, supported by up to 384 sample barcodes, UDIs, with universal truncated adapters, with or without molecular barcodes. Also fully supported on the Avenio Edge truly walk-away automation system. We also have a new innovative technology, single-day workflow, the KAPA HyperPit workflow, based on primer extension target enrichment that combines the high performance of hybrid capture with the speed of amplicon workflows, ideally for somatic oncology research from challenging samples. You can always use the HyperDesign online interface to make use of our optimized probes and primers design algorithms to achieve comprehensive coverage of your regions of interest. And you can always find designs that experts have designed. On our design search space, you will see panels for cardiology, neurology, hereditary genetics, or hereditary cancer. Our latest release is the KAPA HyperExome V2 Probes that comes to upgrade your whole exome sequencing experience by unlocking previously inaccessible genomic regions with our design expertise and extensive optimization. You can stay up to date as the databases keep updating. It provides you the latest databases coverage, such as ACMG version 3.1. You can utilize the KAPA HyperExome V2 Probes with a KAPA HyperCap workflow, supported by the KAPA EvoPlus kit for even higher quality sequencing data, streamlined integrated enzymatic DNA fragmentation. Every lot of the KAPA HyperExome V2 Probes is not just tested by NGS-based probe QC, but it is also functionally tested through capture and sequencing to ensure consistent, reproducible performance in your hands. And you can always utilize the KAPA HyperExome V2 Probes on the Avenio Edge system for true walk-away automation. The result of the improved database coverage and the extensive optimization is shown in this chart, where with the green bars, you can see the percent of bases that are covered by 30 unique reads from the KAPA HyperExome V2 Probes, either with the KAPA EvoPlus kit, the KAPA HyperPlus kit for enzymatic DNA fragmentation, or the KAPA HyperPrep kit for mechanical Covaris-based DNA fragmentation. And you can see how it compares across several important genomic databases, such as Assemble, RefSeq, CCDS, and Cosmic Cancer Gene Census, and how it performs better by covering more bases from the databases by at least 30 reads. So with the KAPA HyperExome V2 Probes, you can cover more bases, see what other supplier exomes may find difficult to see, add more value and confidence in your results, and eliminate more blind spots, with fewer unknowns compared to other exomes. But today we have, as I said, a very important speaker that I would like to introduce, and he will speak about his experience with KAPA HyperExome version 2 probes. Our invited speaker today is Professor Massimo Delledonne. He will discuss about the latest whole-exome sequencing solution that shows high performance in real-world setting. Professor Delledonne comes with more than 30 years thought leadership in genetics and molecular biology. He has an interdisciplinary approach that connects genetics, molecular biology, bioinformatics, and genomics. He is a full professor of genetics at the University of Verona, and he has been a pioneer in next-generation sequencing technologies and in the field of personalized genomics. Without further ado, I would like to welcome Professor Delledonne to the stage. Thank you very much for joining, and the stage is yours.
Thank you, Marcos. And I will share my presentation. I hope you can see it. Yes, so I just need the laser pointer. Okay, so yes, thank you. I'm very happy to be here today. I'm coming from a lovely city, Verona in Italy. Although I don't really understand why it's lovely, because Romeo and Juliet, okay, they are from here, but they both died. Anyway, so I like webinars, but I prefer to meet people in person. At least I can see when they complain, or I can see when they laugh. Here is flat, completely flat. Anyway, what I like to tell you is that I'm coming from a department of biotechnologies. We keep testing and benchmarking applications. In this case, I've been testing the version 2 of the KAPA HyperExome and comparing it to the version 1. I have to say that when Roche proposed me to test the version 2, we defined the date of the webinar well before the data were available. Okay, so this was my condition. I told them, okay, a webinar, but I will show the data that I will get. They were, of course, quite confident in their products because they decided to have the webinar anyway. And today I'm going to tell you something that is, anyway, as you can see, limited to germline analysis. So I'm not going to talk about cancer. I'm not going to talk about coverage of cancer databases and so on. And the data that I'm going to show you today are based on the primary target on AG38. Roche has two designs. One is the primary design. As you can see here, is covering the regions against which the probes were designed. And then there is the capture target that is actually the region where the probes have been designed on. So focusing on primary design. As you have probably, I thought Marcos was showing the difference in the size of the two designs. So the version 2 is bigger. And that's strange because usually the company is trying to shrink the design in order to save in the cost of sequencing. They decided to improve it, increase it, and I think they did something good because, you know, it's very difficult for people also like me to say what is a whole exome analysis. What is an exome? At the beginning, it was just all the exomes. We have about 200,000, 180,000 exomes that are composed in the 20,000 genes. But then there are many variants which are outside of the genes that are clinically relevant. So today it's very difficult to say what is an exome. So by comparing, for instance, the version 1 and the version 2 with the RefSeq, that is a database that many of the hospitals with which we are working, you can see that the majority of the RefSeq is covered. There is a very small portion that is almost identical in size that is not covered. And you see that the main difference is, however, between the V1 and V2 is outside of the REF-seq area. So for us, it's very much important to have all the clinically relevant positions that are covered in ClinVar and HGMD. We analyze the design of version 1 and 2 with the ClinVar, the last version you see, April 2023, and the HDMD Professional 2023. ClinVar has about 216,000 variants, whereas HDMD is reporting more than 368,000. You can see that REF-seq is covering many of those positions, but not all of them. There are about 30,000 variants or positions, clinically relevant positions that are not covered in REF-seq, and more than 40,000 for HDMD. Then you see version 1 of the KAPA HyperExome. There is a better coverage of the clinically relevant position of ClinVar and HDMD. But you can see that in version 2, what they have been doing, it has been further improving the coverage of clinically relevant position. And so the regions that, or the positions that are not covered now in version 2 is much smaller compared to before. Then, if you are wondering what is left out compared to, for instance, REF-seq in those 100 and something thousand nucleotides, well, very little. Actually, as you can see here, there are in version 2 only 4 genes that are clinically relevant, which are in those regions of REF-seq that are not covered by version 2. But not complete genes, just 9 exomes from 4 genes. In those regions, there are only 2 clinically relevant positions for ClinVar and 12 for HGMD. But when we tried to see if we could read those positions on a whole genome sequencing, we found that actually those are very low quality mapping regions, and so it's impossible to read this the genotype of this position. And here you can see the genes that are out in version 1 and in version 2, actually there is nothing out, okay? So what we have done, it has been to take 8 samples that are 2 trios that were coming from the hospital we are working with, 1 single individual, and the NA12878. That is very much important for us because for the benchmarking, we can see true positive and false positive variants in this sample that has been analyzed. You all know that is coming from the Genome in a Bottle project, okay? The 8 samples were processed with the KAPA EvoPlus library, and then they were captured with version 1 and with version 2. And they were sequenced then on a NovaSeq 2 times 150. Library, we follow the protocol and the recommendation of KAPA, 100 nanogram, 18 minutes of, it's an enzymatic fragmentation, clean up 7 cycles of PCR, and the cleanup post-PCR. You can see the 8 samples here. You see that they are very, very similar in terms of size. Actually, running those samples on the bioanalyzer gave us an average size that is almost identical. It's incredible. It's a very reproducible sizing of the fragments that are very close actually to the recommended size of the library, that is 320. And also the concentration was very similar. So the library prep is working very well. Then we sequence 150 in per end, and we applied this bioinformatic workflow that is a classical workflow, BWA and JTK with the haplotype caller. We repeated the analysis also with Dragon, and the results are very similar. But today I'm going to show you those data. First of all, I cannot see you in the eye, so I don't know if you are all expert and you don't like these minutes that I'm dedicating to the explanation of the metrics, or if there is some of you who is a young PhD student or postdoc who is not so well aware of this, so very happy to know this kind of things. But I consider that there are young people there, so I'm going to explain you something. This is a dream situation, okay? So these are the exons or the design, the targets, and these are the reads. You can see that all the targets here are covered, and the coverage is very much uniform. But in the majority of the case, this is not what you observe. You observe something like this. So there are targets that have a very low coverage compared to others, some targets that have an extra coverage compared to others, and some targets like this one, which are not covered at all. So the metrics that we use to describe the quality of the uniformity of the enrichment capture system are two parameters, the fold deity, base penalty, the fold deity, and the uniform of coverage that is 20% of the mean coverage. The fold deity, look, it's the fold of additional sequencing required to ensure that 80% of the target achieve the mean coverage. That means if you have an average coverage of 100x, how much you have to sequence extra in order to bring 80% of your target to that 100x coverage. Usually this value goes from 1.5 to 2. So if you have a fold deity of 2, that means that if you want to bring 80% of your target to the average level of the coverage, you have to sequence double, okay? The fold deity is a good parameter, but is calculated on non-zero coverage position. So that means that in this uniformity matrix, you do not consider those positions that you would capture, but actually you did not capture. For this, there is another parameter that is the uniformity of coverage that is telling you how many positions of your design have a coverage that is at least 20% of the average. In this case, if you have an average of 100x, how many positions of your design have a 20x, that is 20% coverage, okay? Then coverage, okay, we all know that is the number of reads that cover a certain genomic position, but the genotypability, look at this slide, you see here that there are reads that are white. This means that the mapping quality is very low here. These are duplicated regions, you see. Reads can map here and can map here. So those regions are covered, but it's impossible to define the genotype because you don't know where the reads should map properly, okay? For this, I like to introduce the GVCF. Many of you know of it, but probably not all of you. We keep using the VCF file format that has been developed by the 1000 Genome Project. But the scope of the 1000 Genome Project was simply to report variability in our species, okay, variations, not to be able to report the genotype in every single position. So the problem of the VCF is that in a situation like this, the VCF is reporting that here there is one variant, homozygous, very good quality, so it will pass the filter. In this case, there is a position that is identical to the reference, so the VCF is not reporting anything. In this case, the variant is cold, but the quality is very low, so it will not pass the filter. In this case, there is enough coverage, there are not information, so the VCF is not reporting anything. So you see, there is no difference in a VCF file between this position where the genotype is unknown compared to this position where the genotype is known. This is a reference position. And the genomic VCF, that is a little bit bigger than a VCF, introduce the genotype of the reference. Like in this case, you can read in the VCF file not only the variant position, but also the reference position. So that means, are you looking for a variant and has been called? Okay, but if the variant has not been called, is the variant in a position that is pass? If it is in a position that is passed, that means like this one, and it has not been called, that means that this position is reference, okay? why I like to say this, because for us, genotyping or genotypability is much more informative than coverage. You can see here the number of variants which are pass, or number of variants which are passed in position that have a coverage of at least 10 reads. Many hospitals just look at this column here. And you can see that where the pass as well and the pass with a coverage of 10, they soon reach a coverage. We are talking about germline mutation or variation, so no somatic, no mosaic here. Okay? You can see that here there is a plateau that is reached, whereas if you look at the coverage, you see that the coverage keep going up. But if you look at the variant that you can, or the amount of the design that you can read properly, you see that you are already at saturation. You don't need to sequence more or to get an extra coverage in order to call the genotype of many positions. You are already at saturation of 50x. This is version 2 of the KAPA HyperExome. Already with 50, but we will see many more data later. And with the read depth of 10, it's already a 70x. Then on target, near target, off target. Of course, the on target is the one that is interesting for us. For us, the primary target is just this one. But because the fragments are usually a little bit longer, let's say that you have probes in this position of your target, probes are going to capture pieces of DNA, and if the fragments are, for instance, 190 nucleotides long, in this case, you will have a pad or the mapping of 100 nucleotides of the fragment, which is outside of the target region, that is the near target. So near target depends on the size of the fragments, whereas the off target is the background noise that you don't want to see. So people like, if this is your design, you try to keep your fragments short because you want to reduce the amount of data that are outside of the target. You want to avoid the extra tail that is a waste of data and of money, okay? Okay, so the sequencing statistic, we sequenced at 2 times 150, but then we produce also the data by trimming them because many people prefer to sequence in order to, they think, to save money, to sequence shorter, 2 times 800 nucleotides. So same data, these are real, these are synthetically trimmed, okay? Number of fragments obtained, what you will immediately see is the number of duplicates, huge, but I have to say, mea culpa, we say in Italian, okay? That is, it's a problem that we are facing since a few months with the sequencers that we are using, and we get a very high level of duplicates also with PCR-free whole genome sequencer where you cannot have duplicates. The point is we are discussing with Illumina, and in the next few weeks we are going to meet with them because the majority of the duplicates are just duplicate twice. So it may also be that they are the Watson and Crick fragments coming, strand coming from the same fragment. We don't know. So please, I apologize. Don't look at the duplicate number because I'm unable to give you a reliable data about the duplicate rate, okay? Insert size, as you can see, between the two libraries, insert size is almost identical. Insert size is slightly smaller when you trim the read, although you could say that the difference is negligible. And the coverage, you see that HyperExome version 1 has a higher coverage with the same amount of data on 2 times 150. The same is true for HyperExome version 2. So slightly, it's not very much different, but this is due to the fact that so we always consider that the exons are less than 200 nucleotides, 150, 107. So there is no reason to sequence longer, okay? But if you look at the library, and you have a library that is, like in this case, as an average, 140 and 154 nucleotides, you can see that many nucleotides are longer than the 200 nucleotides that you are able to sequence when you sequence 2 times 100, okay? And so if you sequence longer, 2 times 150, you can get more data that when you look at the number of million of fragments that you need for a 60x coverage of the target, you can see that by sequencing longer, you need a little less millions of fragments. Anyway, statistic again, look at the fold 80 penalty. I told you if this number is 2, that means that you have to sequence twice in order to have 80% of your target region at the average coverage, in this case 60x, okay? So this number, when it's very close to 1.5, is very good, okay? In our experience, this is a fantastic uniformity, okay? Then you look at the uniformity that is considering also those positions that are not covered. So this is the difference between the fold 80. And you will see that the uniformity of version 2 is a little bit higher. And you can observe it here because you have the region that have 1x coverage that are a little bit higher, okay? The same data are observed, the same information are coming from the 2 times 150. There are no difference, okay? And if you look at the pass, so the percentage of the target that we can actually read the genotype, we can see that it is in line. Actually, we can say that in version 2, this number is a little bit higher, but consider that version 2 is more than 2 megabases longer than version 1. So that means that the number of nucleotides that you can analyze here properly is much higher. So because the data are identical, in the next slide, I'm going to show only the data using the 2 times 150 data set. Of course, we have all the data also with the 2 times 100, but results are very, very similar. So in this case, this is downsampling, okay, of HyperExome version 1 with reads 2 times 150. The coverage you can see is increasing, but the genotypability is reaching a plateau already at 40x. You see there is a very, very small increase. So you really don't need to go so higher. If you consider the 10 reads of coverage, you start at 60, but you are already at saturation here, okay? In this case, this is version 2. You see this data. We produced this data about 1.5 months ago in a single run. Some samples didn't really reach the expected coverage because of that very high number of duplicates. So if you see that here are only 4 samples analyzed, is because we had 4 samples that we could compare between V1 and V2, but I think it's very good anyway. One of those samples reached 75, so these data are probably very close to the real data, but one sample is not reaching the level, so could have reduced a little bit the number. Anyway, also with version 2, saturation is already at 40x of coverage for the pass. For the pass with the 10 reads of coverage, it's like version 1 at 60. Okay, then variant position that we can read that are coming from the CleanVar database or HGMD. You can see that with the version 1, we can analyze 89.8% of the positions, with version 2, 95.9. So it's slightly increase in the number of variant positions that we can analyze here, the same for HGMD. And you can see that the difference between a 60x coverage and the 80x coverage is null. The numbers here are identical. Then we went to call the variants, and you can observe here that there are about 2,000 variants of difference. So V2 is calling more variants compared to the V1. Of course, it's a bigger design. In this case, you see the variant that have been called on NA12A78. This genome allowed us to see the true positive and false positive of the variant that we call. By analyzing only the single sample, you see that the differences are identical. There are 2,000 different variants compared V1 and V2. And by looking at the true positive variants, you see that those 2,000 variants are all true positive. There is no variation in the number of the false positive. Here there are 2,000 variants more. Here the difference is about 200-300. Okay, then I found this incredible article that is, you know, sometimes on the New England Journal of Medicine, you know, sometimes we as a scientist dedicate times to things that you can consider very silly or useless. In this case, they've been watching all the episodes of ER, Chicago Hope, and the Rescue 9/11 in order to see all the episodes of cardiopulmonary resuscitation, CPR. And they found that at the end, in the majority of the case, the cardiac arrest was caused by trauma, and that the survival rate in the movies, in the videos, is much higher than in the real situation. So you can say, okay, not really important, but look at this. I love this paper. If you are a young PhD student, read this paper that is really fantastic. It has been published 12 years ago, and is counting the injuries that have been caused in Asterix and Obelix. And it has been beautifully written, this paper. You see, in 83% of the case, the brain injury was caused under the influence of a doping agent called the magic potion. Okay, read this paper and you learn how to write very good paper, okay? But you can say, why to do this kind of useless scientist? Well, you know, science is never useless. We wanted to do something similar. Not that it's counting the injuries in Asterix and Obelix, but to try to extend, to go against the flow. And instead of going very short in fragment length for whole exome sequencing, we try to go longer. We know that sequencing on Illumina platform is good up to 500 nucleotides. Then no one knows why, there is a huge drop in the quality of the second read. But up to 500 nucleotides, you can go there. And, you know, there are positions that are clinically relevant in which is very difficult to define the genotype, usually because maybe the exon is duplicate. If you have short reads and short fragments, and this exon is duplicate, the mapping quality of the reads will be very low. But if you have longer fragment, it may be that one of the two mates is mapping outside of the exon, that in any case it's under constraint, so no mutation, no differences between the members of the family. But in the intron part, that the intron is not under constraint, with the time, we had haplotypes that allow us to identify and properly map the read. So when one read is mapped properly, it transfers the quality of the high quality mapping to the other read. And in this case, it's possible to call the variant. This is an example of a variant that is not passing the filters in the VCF because of many low quality reads, and is passing the filter by increasing the insert size. So you may say, okay, by doing this, by increasing the insert size, at the end, you get more near target reads, and so you're losing money because you're sequencing useless data. But actually, if you take the primary design for KAPA HyperExome, but not just for KAPA, you can see that the mean is 186 nucleotides, but the median is 127. So that means that there are many very short fragments, but in order to reach a mean like this, there must be also longer fragments, okay? And if you look at the distribution of the fragments in the regions, you can see that 80% of the regions are having a size up to 200 nucleotides. Only 20% of the regions have a site that is longer than 200 nucleotides, but in terms of bases, the regions longer than 200 nucleotides contains more than 53% of the nucleotides of the entire design. So we repeated the analysis in order to produce longer fragments that were captured also with version 1, but because time is going so fast and otherwise too many data, I'm going to show you only the comparison of the KAPA HyperExome version 2 with the standard fragment size and with longer fragment size. As you can see, this protocol deviation has not been validated or supported by Roche. However, I can tell you that Roche helped us to solve the problem that we had in going longer. So they do not support, not validate it, but they helped us. Now, this is the insert site that we got with the longer fragment. It's more than 300 nucleotides, as we expected. You can see that, as expected, the number of nucleotides on target decreased. This is not identical, okay? These are the raw data, but there is just 1 million result difference. So in this case, you see that there is a huge drop in the number of on-target bases and a huge increase in the near target, of course. The coverage, however, you can see that despite the fact that there is a drop in the on-target, there is an increase in the coverage. And why is that? Because also in this case, you see, this is the average of the fragment, 315, but there is a huge tail that contains many useful data. And so to reach a 60x coverage of the primary target with the standard library, you need 29 million fragments, whereas with the long fragments, you can save 8 million fragments, that is 16 million reads, okay? Comparison 60x and 80x, you can see that the default 80 penalty, very similar, a little worse on the long library that we improved very much, thanks to Roche, but it's not still perfect, but the uniformity is higher. The uniformity is higher, and this is good because this allows us to have a very good evaluation of the data that we get. This is the forgot the name. Oh God, what is this? Okay, you see forgot the name. No one can suggest me. No. Anyway, so you see that over here, by increasing the coverage, you reach a saturation. Downsampling. Oh, downsampling. Downsampling is showing that also here, for version long, we have identical data, saturation of 40x for the pass and 60x for the pass 10x. Then the number of reads passing, the number of reads, the percentage of the design that is passed, look at the difference here. The longer fragment has a higher level, both at 60 and 80x coverage, 1% of difference in the number of pass position. And in terms of variance, you can see that the variants are very similar. Actually, you can say that there is slightly increase in the long fragments, but the difference is really negligible. But when you go and check for the true positive and for false positive variants on the NA12A7A, you still see that there is this very small difference here and this very small difference here. So probably there is no reason to really extend the fragment side in order to capture 1 or 2 few hundred variants more. But what is very much important is that if you are interested in some genes that are usually not totally characterized, like P10, for instance, you see that with the short fragments, you can get the genotype of 85% of the gene. Well, you can get the genotype of 100% of the gene by simply increasing the fragment size. And in terms of rep-seq genes having 100% pass, with a longer fragment, you increase of about 900 genes, the genes that are completely analyzed. Then, I'm very close to the end. The cost of the sequencing. Well, I'm showing the data just with the NovaSeq, but here I reported the SP, S1, S2, and S4 for sequencing 2 times 100, 200 cycles, or sequencing 150, twice 2 times 150. You can see that the cost per gigabase is lower when you sequence longer for the SP, for the S1, for the S2, and for the S4. But the cost per gigabase, per million fragment is a little bit higher. So if you sequence longer, the cost per gigabase is lower, but the cost per million fragment is a little bit higher. But now let's see how many million fragments you need to sequence and reach a 60x coverage by sequencing 2 times 100, 2 times 150 with the standard or with the long library. And you see that there is a quite dramatic difference in the number of million fragments that you need, okay? Now, if this is the table, and here you report the number of million of fragments multiplied for the cost per million fragment, what's come out is that if you sequence short, actually on a NovaSeq, you spend exactly the same amount of money if you sequence 100, 2 times 100 on 2 times 150, in order to reach 60x coverage. So this is very much important because sometimes you want to use a smaller flow cell because you cannot sequence your exome together with other samples that require 2 times 150. So it's much better to try to go on an S2 and save money here, okay? And sequencing 2 times 150. Then if you go long, actually, you see quite an important reduction. So by sequencing longer, the longer fragment, you save much money with the SP, with the S1, with the S2, and with the S4. Then, if you compare version 2 by considering sequencing with the standard fragment length 2 times 100, or sequencing longer fragment 2 times 150, you see that there is a quite important reduction for all the possible flow cells that you want to use. Well, here are my conclusion, very simple. I think that both the version 1 and version 2 of the KAPA HyperExome have a very good enrichment uniformity. KAPA HyperExome V2 is a little bit bigger, but this is a very good thing for me because it has a better coverage of clinically relevant position. If you want to save money, don't stay with version 1 because it's smaller, simply improve your ability to sequence on a more powerful flow cell that the cost per gigabase is lower, and then that at least for us, increasing DNA fragment size and sequencing cycles, so going to 2 times 150, increase the coverage, increase the genotype ability, and decrease the cost. Then, finally, I like to thank my collaborators, my group, in particular the people from the wet lab, Chiara and Antonio, who produced all the libraries and the sequencing, and Luca and Matteo, who spent their last month of life working around the clock in order to provide me the data. The last slide has been edited at 3:30 today, so just 1 hour and a half before of this webinar. And of course, my collaborators, the help from Emanuele and Marcia from Genartis, and our collaborator from the May Hospital, in particular, Angela Peron, Rosangela Artuso, and the entire group that allow us also to make this kind of testing, and then they take the result that we produce and implement them in the clinics. And of course, I like to thank you for your attention. And I'm ready to take questions, if any.
Thank you very much, Professor Delledonne. Just let me introduce myself shortly. My name is Xavier Miró. I'm a senior application specialist in Roche Sequencing customer support, and I will be taking care of the Q&A session. please do not hesitate to use the tool for your questions. And I will be checking. Remember also the questions can be ranking. So the questions that you prefer to be placed first will be ranking up in that tool. Actually, I have some questions already in that tool. The first one, it's referring to mitochondria. Do you also check the mitochondrial genome for variants?
There are 9 genes in RefSeq that are related to mitochondria genes. We excluded them from this analysis. So the answer is not, I did not check yet.
Mm-hmm. Okay, maybe to comment that, taking advantage of this question, that in case that one lab would be interested also to capture mitochondria, we decided not to include it in this design. So there is a specific design for mitochondria that can be spiking. So it means that you would have your HyperExome version 2 design, and then there is another design just for mitochondria that you can spike in, and then following our support, you will be able to get also the information for mitochondria. Thank you. The second question, it's referring copy number variants. Did you also detect them? Were you interested to monitor them?
Not for the moment. Frankly speaking, we didn't have the time to analyze everything, and we still have to do that.
Okay, so it's something in plan in your case. Excellent. Also, one question I think interesting because it clarifies the workflow. The question is if Evo Plus, the option that you use, it's using mechanical or enzymatic DNA sharing.
Well, we like, I'm an old person, I prefer the mechanical one, but I have to admit that in the last few years, the enzymatic system improved very much. And the quality that we see now, and the protocol is very easy. What lab people tell me, they like the protocol, they say the protocol is easy, and as you can see, it's very reliable in terms of size. So I like the enzymatic one.
Okay, maybe just to clarify from Roche side, actually there are 3 options. One is Evo Plus, the one that you selected, that it's enzymatic fragmentation. There is also HyPA Plus, that is also enzymatic fragmentation. And then there is the option of using HyPA Prep, that would be the mechanical fragmentation. Actually, the 3 options are included and supported in our instructions for use. Actually, the next question refers to your experience. So it's in the comparison between version 1 and version 2. It's what is the biggest difference between version 1 and version 2 for you, Massimo?
For me, it's the big difference. What I appreciate is that version 2 is bigger. So it's the time of shrinking the region that we analyze over. The cost of sequencing is now so low that it's much better to increase the regions of the genome that we analyze. So version 2 is much better because it's covering many more clinically relevant positions. And because of this, I consider the version 2 much more informative than version 1. So the pros is this one. The cons is that it's a little bit more expensive because you have to sequence a little bit more. But as I've shown you, the big difference at this point is not 1-2 million reads, it's on which flow cell you can sequence. That is making a huge difference in the cost.
Yeah. So as we were remarking during your presentation that the version 2, it's bigger, right? So actually the primer target, right? It's bigger, but we were able to keep a capture target that it's quite close. It's quite similar to the version 1, because as you were highlighting, depending of the sizes and several aspects will increase the sequencing cost clearly. And I think from that point of view, I think that you gave a very good example. And maybe I personally, I'd like to ask you maybe to go deeper about the metric that it's the uniformity, right? So how important is the uniformity regarding not adding extra sequencing costs? You gave the example of quality based penalty of 2, that would mean to double the sequencing cost in that case. Maybe you can give us some your general experience about how the uniformity at the end is increasing the cost.
Well, for me, uniformity is very much important because, you know, if you have some regions with a low coverage, you may not call the variant there. You get the genotype, but you get the wrong genotype because you may have a allele imbalance. And so if you have a very poorly uniform region that is reaching your threshold, but is not reaching such a high coverage like 50, 60x, you may not call a variant there. If you have seen the data on the false positive variants in version 1 and 2, you see that the false positive are very low. And anyway, there is not much difference between the number of false positive with low coverage and high coverage. And this because the uniformity is very good. If you would repeat the analysis on an enrichment system that is very poorly uniform, you will see a huge variation in the number of false positive and false negative in the downsampling analysis, okay? So here the quality is good, and I would say having a good uniformity, if you have to buy a kit, better if you buy a kit that is giving you uniformity than buying a kit that is not giving you uniformity. And then you see saturation, you reach plateau in the variant calling. It's not that the number of variants called on NA12878 is dramatically different from 30x to 80x. It's the same number of variants. So when you have a good uniformity, you don't need to reach a very high coverage with germline, not with mosaic and somatic mutation, of course, but this is another story.
Okay. Excellent. Another question is regarding the T2T or the telomere to telomere genome that we have been using in this case when we have generated the HyperExome version 2. Maybe let me explain for the audience what is the T2T. The idea is that there are some regions in the AG38 that still the information is still limited, and actually there are a high copy or high repetitive regions. And when we design the probes against those regions, what they will bring is to more unspecificity in the capture. What we did is in our design for the version 2, we included this T2T or this telomere to telomere that provides us more information about these repetitive areas. And this allows us to be designing probes that they will be more specific and to avoid these ones that they just will be capturing more repetitive regions and bringing more unspecificity. I just did this introduction, Professor, just for the audience, but maybe they are not familiar with this T2T. The question is, it's important for you, did you use T2T genome for the alignment?
I would say yes, but I would say that by reading the last Nature paper on the human pangenome, seems to me that using the pangenome is even much better than using the T2T genome, okay? So we can also always improve. For me, it's already very much important that I could perform the analysis on AG38. Many of the hospitals I'm working with still require the data on 19, HD 19. That is so old. So yes, I hope one day they will move to the telomere to telomere and to the pangenome, but I really suggest them to soon forget about 19, at least to embrace the 38.
Yeah, very good suggestion. The next question is regarding also going back to the workflow. How long was the hybridization reaction in your case?
Oh, you know, I don't know. I have to ask my wet lab people who are in another room, not here. But we have been following exactly the protocol. So maybe some of you can, from Roche can answer.
Well, I can take it. So classically, in our procedure, we recommend the overnight hybridization because we know that this will bring you the best metrics and also will play a role in the uniformity. But we are also testing shorter hybridization times. And then basically what we observe is that the shorter the hybridization time, lower the uniformity. So with the time, the uniformity is increasing, and because uniformity is an important metric, then we suggest this overnight. But anyway, it could be that for some application or for a concrete lab, to speed up the hybridization process would be relevant or important. and then having a shorter hybridization reaction could bring already the uniformity required and be also a good solution for that lab. So we prefer to offer what we know that rents the best uniformity, that is the overnight. But in case that someone would like to shorter, we have done some tests, not for the version 2 already, but we know that in some cases, shorter hybridization times would be helpful. So rather than modifying this protocol, we try to provide information how the time impacts, and then the labs can decide if it's convenient or not for them. Okay, I go back to the tool. Checking, I think that there are no more questions that I cover. We'll just
perfect in time because 5:56. Perfect.
Exactly. Just checking that it's nothing that arrived at the last moment. Anyway, if someone places a question and would not be covered in this call, we'll address it and we'll go back to you with the answer. No worry for that. No question will be missed. Okay, so then if there are no more questions before closing the session, I want to thank you, Professor Delledonne, for your participation. I would like to ask you if you want to add anything from your side before we close the session.
Sorry, I didn't understand what.
Do you want to add anything else from your side, or some goodbye sentences?
I was happy. I like when companies allow me to test their product and blindly organize a webinar and allow me to describe the product, considering that if the results are good, I say good things. If the results are not good, I say not good things. So, so you guys took the risk, and but you knew that your product was good. So thank you for having allowed me to test it and also to share with the audience the result.
Then on behalf of whole Roche, I want to thank you again, Professor Massimo Delledonne, for your great contribution with this webinar and for kindly sharing with us your experience and conclusions with the Roche KAPA HyperExome version 2. We really appreciate it, and it helps us to understand better your needs and to continue in our innovation path. And obviously, I would also like to thank you very much, the whole audience, for your time during this webinar and for your questions. We'll send you a very short survey to understand how did you find it, what topics do you find the most interesting ones, and which content brings you most value. So then thanks again and have a nice rest of the day. Thank you very much.
Bye.